Okay, so welcome back everyone. Today we're going to be continuing our study of protein structure by looking at the topic of structure searching and alignment. Essentially trying to do something analogous to what we do with sequence searching and alignment, but in this case looking for structures that are related to some piece of biological data we have available already. So last time we had seen a bit about how we can find structure. So if you have a new protein, a new molecule that you're trying to discover a structure for, we have various experimental options, some computational options we can apply to find a structure, but all of these have some significant drawbacks. The experimental options tend to be a pretty significant amount of work and often won't succeed on any given protein. The computational options are not nearly as much work, but are often going to do a poor job, so not really reliable. Often a much better way to find a structure related to a sequence that we're interested in is to try to take the sequence, or maybe even an existing structure, and try to find structures we already know something about that are similar to the one we are looking at. And we're going to see two variants of this in the course of this lecture. In one case, we're going to be looking for the topic of looking for structures given a sequence. So in other words, given a biological sequence, find structures that might correspond to the structure of that sequence. And that will often be done in the context of looking specifically for protein domains that may correspond to that sequence. We'll be seeing some methods for that. And I'll spend less time on the topic of taking a structure and finding related structures, although that's also something we might want to do. I'll be spending less time on it primarily because the methods are not as well developed and not as reliable as searching for a structure of the sequence. It's also something we're less often going to need to do, but we'll see a bit about how both of these things work. All right, so to begin, finding protein domains, or in other words, searching for structures given sequences. So a very common way of doing this is to look for sequence motifs. And I've used this term before in previous contexts. A motif is simply a pattern that identifies a piece of sequence, often has some functional significance in a piece of sequence. And in the context of protein structures, a sequence motif would typically be important because it's something that is characteristic of a class of protein structure, typically a domain structure. So there are some basic ways we'll go about trying to find a motif, and most of them come down to a relatively simple set of basic operations on which we can see a few different variations. And for the most part, what this begins with is, first of all, trying to find representatives of the class of structure of interest to us, and then building a mathematical model that would let us represent or identify new members of that class. So, for example, what we will typically begin by doing is taking a bunch of sequences that we know have the structure or the structural domain of interest to us, and performing a multiple alignment to try to identify the specific amino acids or residues that line up between different examples of that particular sequence pattern. And then we'll build a mathematical model known as a position-specific scoring matrix, or PSSM. You'll sometimes also see the term position-weight matrix, or PWM. Basically, that's the same thing. And this will be a mathematical model that tells us how likely a given new sequence is to be a representative of the class of sequences used to build this PSSM. And we'll look in some detail about how we build these and how we use these in practice. I'll point out that this is something I'm covering now in the context of identifying protein structural domains, but this basic technique is used very widely in sequence analysis. It has a number of other important uses we'll be revisiting later in the term in other contexts. So just be aware this is something that comes up again and again as a particularly useful kind of mathematical tool and abstraction for working with sequence data. All right, so the basic idea is that we have done our multiple alignment, so we might imagine we've got a set of different sequences, each of which is a representative of this class of protein structures of interest to us. And what we're going to want to do then is build a mathematical model that tells us what the members of this class have in common so we can better recognize new members of that class. So, for example, we might imagine that this is a conserved region of amino acid sequence, in this case a set of 8 different proteins that all have this structural domain of interest to us. And what we would then want to do is pretty much just run through the positions in this multiple alignment and ask what are the biases, what are the frequencies with which we observe particular amino acids in those positions. So, for example, we could take this position as one example of this, and we could simply ask, how often do we see particular amino acids in that position of this multiple alignment? So we might observe, for example, that very often we see an S, a serine, in that particular position of the multiple alignment. And that would then tell us that there is a high probability that sequences that have a serine there might be an example of this, or at least a reasonable probability, and things that don't have a serine there are maybe less likely to be examples of this particular protein structure. And we can codify that by asking specifically what is the probability of a serine in that position. And we can empirically estimate the probability based on the observed sequences to be 5/8. So we have 8 examples, 5 of them have a serine in that position. Similarly, 2 of our 8 examples have a P, a proline in that position. So we say the probability of observing proline there is 2/8. 1 has an alanine, so probability is 1/8. And then we would have various other amino acids that maybe aren't observed at all, and we could say those have a probability 0/8 of being observed in that position. So that basically is the idea behind a PSSM. We would run through each of the positions in the conserved region of this multiple alignment and do this kind of assignment of probabilities to those positions. In practice, things get a little more complicated than that, though, because if we do this exactly as I've described it here, we'll actually run into a problem, especially if we don't have too many examples, and that is that it will become hard to recognize new examples if they just happen by chance to be different from our observed examples. So in other words, we would conclude from looking at this position that you never see a cysteine in this position. But it may be that if we had looked at 100 sequences instead of just 8, maybe 1 out of 100 does in fact have a cysteine in this position. And if we want our model to be able to have any chance of recognizing those examples that differ from the things we've already seen, then we're going to need to throw in some additional kind of flexibility here, so more or less flatten out our probabilities a bit so that we have a chance of identifying things that aren't in our examples. And one simple way that's often done is to use what's called a pseudo count. And a pseudo count is basically just pretending that we've observed extra examples that have amino acids that aren't observed in our real examples. So in other words, we might pretend that we have an extra count of epsilon of each of our positions. So we throw this out and we pretend that instead of observing 5 serines in this position, we're going to pretend that we have observed 5 plus epsilon serines, for some small number epsilon. And that is allowing for some little slack that could account for the possibility that maybe there are other things we haven't observed there. We would likewise say that we observed 2 plus epsilon prolines, 1 plus epsilon alanines, but the really important one is that we will say we have observed 0 plus epsilon cysteines. So in other words, we're pretending there's this little artificial count of sequences in the universe of all possible examples of this domain that have something we didn't observe in our finite set of actual examples. We have to control for that in our denominators. So if we're assuming we've observed an extra epsilon count of each of the 20 amino acids, then instead of saying we've observed 8 examples, we'd say we observed 8 plus 20 epsilon examples. So if we throw in some numbers here, let's pretend epsilon is 0.1, so we've observed an extra 0.1 examples of each amino acid. Then instead of saying we observed 5/8 fraction of serines, we would say we observed 5 plus 0.1, or 5.1 over 8 plus 20 times 0.1, which is 10, or in other words, 0.51 fraction. So 5/8 is 0.625. That would be the actual empirically observed frequency. We're going to, by flattening this out, instead say that we actually think there's a 51% chance of observing a serine there. So we're lowering the probability of that we think a serine occurs in this position. Likewise, we would do something similar with proline. We're going to kind of flatten that out a bit, say that we've observed 2.1 over 10, or 21% chance of observing a proline. But again, where it becomes important is in the rare ones, the ones that are underobserved. So with a cysteine, instead of saying our probability of observing cysteine is 0%, we're now saying our probability of observing cysteine is 0.1 over 8 plus 20 times 0.1, or 0.1 over 10, or 1%. So we're now saying that we think there's a 1% chance a cysteine might occur in this position. Now these pseudo counts are a bit heuristic. You can put some probabilistic theory behind them to say what we actually mean by a pseudo count, but more or less it's a heuristic. It's a trick you throw in to make things work a little more robustly. And to get this to work, you typically want your epsilon, you want your pseudo count to be something on the order of 1 or a little lower. So 0.1 would typically be a good pseudo count, maybe 1 if you've got a reasonably large actual count. You don't want it to overwhelm your actual data, so generally you want to keep it in that range. But let's just say we have done this. We've got our pseudo counts, and we'll imagine that we have run through all the conserved positions of our multiple alignment and got these pseudo counted frequencies. And that set of frequencies across the positions of our multiple alignment is what we refer to as a PSSM or a PWM. Okay, so questions? All right, so we will then suppose that we have done this and we've got a PSSM. And what I now want to go through is what we actually do with this. So you can think of a PSSM as something that describes a way of generating possible new examples of our sequence. So we put together this PSSM, and we would have a set of amino acids and a set of positions, and for each position, we would say a probability density of possible amino acids in that position. So in position 1, we have a relatively flat, not exactly uniform, but relatively close to uniform distribution of amino acids, or maybe 1% of the time we observe alanine, 20% cysteine, so not quite uniform, but we might imagine we've got a decent chance of finding any amino acid. Other positions might be much more specific, so maybe position 2 has to form a disulfide bridge in this particular domain. So there's a really strong evolutionary pressure to keep cysteines in that position. So maybe that position is very specific. It can't tolerate a lot of variation. Others may have more restricted classes of variation they might be able to tolerate, and so forth. But you could imagine that this is something you could use to generate sequences that look like examples of the domain we got from a multiple alignment. So we could pick an amino acid for position 1 from this probability density, pick one from position 2 for this probability density. This one maybe could be a lot of different things. This one would almost always be cysteine, and just run through and generate a random sequence. And you could do this over and over, and if you did this many, many times, you would see more or less any possible sequence occurring with some frequency, but sequences that look like examples of your protein domain are going to occur with much higher frequency than ones that don't. Now, using this to generate hypothetical sequences is more or less a thought experiment. That's not really what we want to do with this, but it helps us understand what we actually want to do with this. And what we actually want to do with this is take observed sequences, some new sequences, and ask if they look like examples of the domain. And the way we are going to be saying that in a probabilistically rigorous sense is to say that we are looking at the quality of a new sequence with respect to this PSSM in terms of the probability the PSSM would produce that sequence. That in statistical or probabilistic terms is known as a likelihood. So the likelihood of generating the data contained in a particular sequence from the model contained in this PSSM. And so we could take a particular sequence, let's say GCEGDA, and we could talk about the likelihood of generating that sequence from this model by simply going through each position and accumulating the probabilities of the given amino acids we actually observe in the given positions. So we could say that G in position 1, we look up, we see G glycine has a 0.15 probability of being in position 1. We could see that C in position 2 has a 0.9905 probability, and so forth. And the product of all of these probabilities is then referred to as the likelihood of this sequence being generated from this model. And that, more or less, is how we're going to decide if we're looking at a good sequence. So we might look at this and decide that this is a pretty good likelihood. So maybe this is a reasonably high probability. There's more that'll go into that shortly. But maybe we think this is a good example. And we can say the probability is relatively high of this. There's a high likelihood this sequence comes from this PSSM. Now you could do the same sort of thing with the sequence alignment tools we've seen previously. So we could align this sequence to examples of this domain and ask if it looks like they have good alignments. The reason we would do a PSSM rather than just looking at alignments to known examples of this is that the PSSM is generally going to do a better job of capturing what's important to this specific family of proteins, so this specific structure. So, for example, we might have another sequence that looks very similar to this one, but position 5 is changed. So maybe instead of a D in position 5, we have, I don't know, let's say an E in position 5. In this case, the model would be able to tell us that that's not a very important difference for this particular protein domain. For others, it may be more of an important difference. So maybe having a D or a G would be a very big difference for some classes of sequence, not others. A sequence alignment wouldn't usually have a way of figuring out that this is a sensitive position. This is one where it really matters that you have a good match, others are insensitive. On the other hand, let's say that we flipped this amino acid. So we had exactly the same sequence, except that instead of a G in position 4, we have a D in position 4. The PSSM could recognize that position 4 is a really strongly conserved position. It really needs to be glycine here. Maybe there's some functional reason why it has to be a glycine in that position. And so we could recognize that even though there's just a single base different, it's making a huge difference in the probabilities. So when we do the product of these, we end up with this sequence having a likelihood that is lower by a ratio of 0.005 to 0.9905. So orders of magnitude lower. And we might then conclude that this is not a good example of the PSSM. So that basically is what a PSSM is doing for us. It's capturing the things that are important and the flexibility that is apparent in the multiple alignment of things we know are examples of this specific domain. Okay, so questions? All right, so I mentioned that there are complications here, and one of the big complications is that it's not obvious looking at the number that comes out of it, the likelihood, whether we're looking at something with a good likelihood or a bad likelihood. If you're looking at longer motifs, your likelihoods go down. It may be that in some cases, a likelihood of 0.001 is really good, in other cases, the likelihood of 0.001 is really bad. So one of the things we need to be careful about is that likelihoods and the probabilities we get from these PSSMs are always relative. You can only talk about a likelihood being good relative to another likelihood. And in particular, we would generally be talking about the likelihood of this sequence relative to one model versus the likelihood relative to another model, or more often a library of possible models. So it might be, for example, that we have gone through lots of different classes of protein domains that we think might be present in our sequence. And we have identified that maybe there are dimerization domains, and we care a lot about those. And maybe we've got classes of those. We've built a PSSM, and there are hormone binding domains. We may have classes of those, and we built another PSSM, and other kinase domains, and so forth. So we would build a PSSM based on a multiple alignment of each of these. And then what we would be doing is not just looking at one likelihood, we would be running our observed sequence, the one we were trying to classify, through many PSSMs and comparing the likelihoods that come from all of those. And we would be trying to identify typically what is the maximum likelihood. So what is the best likelihood we observe from all of these models. We would also generally compare this to what is called a background model. And a background model basically accounts for everything for which we don't have a well-defined PSSM. So if we look at the universe of all possible proteins, there may be 100 different domains we know about and care about and have PSSMs for, but most of what's in those proteins is not going to be in any of those domains. And so you build a background model, and a background model is basically something where you would look at, on average, how often do you see each amino acid, and then pretend that every position in the motif you're trying to assign has that particular background frequency, and then just take the product of those, and that gives you a likelihood for that sequence in the background. And then what we would generally do is run through our lot of those and say that that is our best assignment of the classification of this protein, either to a domain or simply declaring that it doesn't match any domain we know about. Even that is oversimplifying a bit, though, because we have to throw in one more bit of detail here, which is that some kinds of protein structures are going to occur more often than others. So it might be that a particular kind of dimerization domain of interest to us occurs very often in sequences. Lots of sequences dimerize. There are different ways they do this, but some of them are used over and over. So it may be that a dimerization domain is really common, and we would want to build that into our analysis. So if we see that dimerization domains are very common and hormone binding domains are very rare, then we would want to take that into account and say, if our sequence looks to be about equally good a match to a dimerization PSSM and a hormone binding PSSM, then really we would want to say that dimerization is the most likely assignment, because dimerization occurs more often, we believe, in the universe of proteins from which our sequence is drawn. So that is captured in the notion of what's called a prior probability, and a prior probability is literally just what we think we know before we look at our data, our prior expectation of the frequencies of these different kinds of domains, or could be the frequency of the background, so basically how often we expect to see something that is not any of those domains. So that might be something we can infer from other analysis of our data, so an independent analysis might be something that just comes from what we think we know about our sequences. But in the end, what we really are going to do here is use the PSSMs like I mentioned, and simply multiply the likelihoods, that is the probability of the sequence given the model, by the prior probability of the model. And that's really going to be the score we assign to our sequence with respect to each possible model. And we would then run through our set of possible models that we have available, through our background model, put all of that together, and the highest probability assignment is the thing that we will say is the most likely structure or lack of structure for our observed sequence. All right, so to make that a little clearer, let's look at an example of that. And to keep things relatively simple, I'm going to pretend that we have a library that only has one domain in it. So we've got one kind of domain, and I will just assume that this occurs relatively rarely, 0.001% of the time. So we've got that domain, and I'll imagine that we've built a PSSM that recognizes that domain. And we also have a background. So we have some background frequency of amino acids we've built for things that are not examples of the domain. And what we would then want to do is take some new piece of sequence, and we would ask the question, what is the most likely assignment? And for this toy example, since we only have one domain in our library, there are only two possible assignments. But either it's an example of the domain, or it's an example of the background. So let's say this is our sequence, CCE, GCG. The first thing we would want to do is run through our PSSM and get an assignment of that sequence, of the likelihood relative to that PSSM. So we take our sequence, we look up in our table, we look at cysteine in the first position, it's got a frequency of 0.2, cysteine in the second position, 0.9905, glutamate in the third position, 0.55, and so forth. Take a product, and we get, in this case, 1.6 times 10 to the minus 4th. So that would seem to be a decent probability. And maybe we would then run through our background. So we would look at the same sequence, and we would say, in our background, what is the probability of a cysteine in any given position? You can see it's 0.02. What is the probability of the cysteine in the background again? 0.02. Probability of glutamate in the background, 0.04. Probability of glycine in the background, 0.1. And so forth. Multiply those through and get a probability of generating the sequence from the background of 3.2 times 10 to the minus 9. So superficially, we might believe, based on looking at these, that the most likely assignment is that this is indeed more likely to be an example of our PSSM model than an example of a background sequence. But we have to add in our prior probabilities. So our prior probability of the motif or the structure that is represented by the PSSM I've declared to be 0.001. And if we multiply that through, what we're going to end up with is, it's not quite right there, but okay, so those probabilities aren't quite correct, but let's say 1.6 times 10 to the minus 9th. And if we multiply through by the background frequency, we believe that the background is occurring 0.999999 fraction of the time, almost 1 fraction of the time. We get a corrected likelihood that was basically the same as what we had here, 3.2 times 10 to the minus 9. So after throwing in that correction, we might conclude that indeed the background is the more likely explanation here. So in other words, after correcting for the prior probabilities, we might conclude that even though the sequence looks like an example of the PSSM, it is actually more likely to be an example of the background. The more likely hypothesis is that it's just random chance that this thing happened to look like an example of the PSSM. So that basically is the idea behind how we would use a PSSM. And of course, in real practice, a lot of this will be automated. We may be working with already existing libraries of PSSMs, but this is the kind of reasoning that goes into them, how they're put together, and how they're used. So, get that. So PSSM is one of the main ways of doing this. It's often the simplest, the easiest one to do by hand. But there are other kinds of models that have greater power, more flexibility for handling more complicated kinds of structural patterns or sequence patterns. And I want to go through another example of this kind of model. And that's something called a hidden Markov model. So a PSSM is going to be something that will come up quite a lot in our later analysis. We'll see those being used to detect other kinds of sequence motifs. And the same is true of hidden Markov models, or HMMs. HMMs come up repeatedly as a tool that we'll use for analyzing sequence data, and this too we'll be seeing in other contexts at several points later in the term. And these, unfortunately, are somewhat more complicated. We're not going to be able to get through in this class at the same level of detail of understanding of how these work as we get for PSSMs, but I think it is important to know what these are, what they can do for you, and why they might be appropriate in some contexts in preference to PSSMs. So an HMM is something you can think of as sort of similar to the PSSM in that you can think about it as an abstract mathematical model or a virtual computer even for generating random sequences that look like an observed pattern. So an HMM has a more complicated mathematical structure. It's typically represented by a computational or mathematical object called a graph, which is a set of nodes, often represented by circles, and edges, which would, in this case, typically be represented by arrows pointing between nodes, and then various other probabilities and other mathematical features associated with this graph that essentially describe probabilities of moving between different nodes, or also known as states of the model, and probabilities of producing different nucleotides or amino acids, depending on how we're using this, in different states. And more or less the way this is going to work is that we would imagine that we are generating sequences by starting in some node or state and then jumping between different states with probabilities of jumps based on these probabilities annotated on the edges. So if we're in this state, then we would have a probability of jumping left that is 50%, a probability of jumping right that is 50%. Every time we make one of these jumps, we're going to assume the model spits out an amino acid. So if we are in the center position here, it has a probability distribution of spitting out amino acids. And in this case, it has a 100% probability of spitting out 8 alanines. So every time we land in this position, we're going to imagine you spit out an alanine. If we go left, we're going to imagine that this one has a 100% probability of spitting out glycines, and maybe this one is more complicated. So if we jump to the right, we're going to have a 50% probability of spitting out threonines and a 50% probability of spitting out cysteines. So basically that's going to give us our model of how this particular HMM behaves. And this is going to capture some pattern of sequences. So it's going to be a way of generating random sequences, but random sequences with some regularity to them. And we can walk through a few steps of this model to see how that might work. So if we imagine that maybe we start in this first position here, so we are in the state shown by this position here, then we will see what the output probability is. We see 100% probability of outputting an A, and so that's what we do first. We spit out an A here. We then effectively flip a coin. We look at the edges projecting out of here, the arrows pointing away from that center node, and we can see that there is a 50% chance of going left, a 50% chance of going right. So effectively, we flip a coin to decide which to do, and maybe we end up going right. We then see that if we've gone right, there are two things we could output. There's a 50% chance of a T, a 50% chance of a C. So we flip another coin. If it's heads, we'll output a T. If it's tails, we'll output a C. Let's say we got heads and we've outputted a T. We then look at what we're going to do next. We see that there is only one transition out, so 100% probability of going left. And so the next thing we're going to do is go left. And when we go left, we end up back in this state. We have a 100% probability of outputting an A. So we output an A, and then we would once again see what our possible transitions are. We could go left or right. We have equal probability of each. So we flip a coin. If it's heads, we'll go left, or it's tails, we'll go right. Maybe in this case, we went left. And we saw that when we end up in this state, 100% of the time we output a G. So that basically is the process. We just do this over and over again, and this is going to generate random sequences according to this model. Now, every time we run this, we might have a different sequence we generate. Obviously, there's a finite set of possible sequences, unless we're looking at an infinite string of them. But some sequences are going to be much more likely than others. So the structure of this model is going to guarantee certain kinds of regularity. The way this is set up, you might notice that it's guaranteeing that every other residue is an A. It's impossible to get any sequence out of this that doesn't have that property. Likewise, between the A's, half the time we're going to have G's on average. Doesn't mean that any particular base is guaranteed to be, or any particular residue is guaranteed to be G or not a G, but half the time we're going to have G's. And when we don't have G's, half the time we have T's, half the time we have C's. So we could, in plain English, come up with a description of what kind of sequence this produces and the probability distribution. But the HMM gives us a nice, compact mathematical model that encodes this sort of pattern and can encode much more complicated kinds of regularities and patterns in these sequences that we would have a hard time describing in plain English. But that basically is what an HMM is. And just like with a PSSM, we can think of this as a way of generating random sequences, but really what we want to do with this is take non-random sequences, so take sequences we've actually found, and ask the question, is this a sequence that looks like it could have been generated from this HMM? And we'll do this qualitatively in a very similar way to the way we do this with PSSMs. We will say that the goodness of a sequence relative to an HMM is the likelihood of that HMM generating the sequence. So the likelihood of this sequence is the probability that if we ran this HMM, this would be the sequence it would generate. And that likelihood is again going to be the thing we use to evaluate a sequence. So to give an example of how that might work, let's look at a real protein motif we might study with an HMM, and that is a coiled-coil motif. So a coiled-coil motif I've mentioned before briefly. So a coiled coil is basically a pair of alpha helices that pack against one another and wrap around one another. It's a common way that proteins oligomerize, the different polypeptide chains get tightly bound to one another, and it happens to produce some particular regularities in the sequence. In particular, these coiled-coil alpha helices pack a little more tightly than your standard alpha helix, such that effectively they have 3.5 amino acid acids on average per turn around the helix. So each time we make a turn around the helix, we're going through 3.5 amino acids. And an implication of that is every 7 amino acids are making 2 turns around the helix. And because of that, we can assign positions to the steps along these 7 amino acids in 2 turns of the helix that give us a way of describing regularities in these sequences. So in other words, if we pick a starting point where one of these helices is packed against the other, we can label that position A, and the next amino acid would be in position B, C, D. Halfway from D to E, we would have made 1 turn around the alpha helix, then we complete our turn to E, F, G, and then the next one would be back to A, and we would be back to the same position rotationally around the helix, just 2 rungs further along. So that creates a particular pattern known as a heptad repeat, where you can more or less think of there being 7 different functionally distinct positions within a coiled-coil polypeptide sequence, and then the coiled-coil just repeatedly cycles through those 7 positions. And these positions have different biochemical constraints on them, so different structural constraints that will show up in the patterns of amino acids you tend to see in these positions. So, for example, the A and D positions of one coiled coil will tend to be packed against the A and D positions of the other alpha helix in this coiled coil. So if you have 2 pieces of protein surface packing against one another, that tends to squeeze out the solvent, so it tends to squeeze out the water, and that is going to create some biases in the amino acids in those positions. In particular, it's going to mean that the A and D positions will tend to be biased to favor hydrophobic amino acids, so amino acids that like to be away from the solvent. And so you will have a high bias towards hydrophobic amino acids in the A and D positions of every pair of turns around these helices. The others will have some generally weaker biases, but generally that bias towards hydrophobics in the A and D positions are the most obvious and dominant thing that helps us recognize these. And we can build HMMs that recognize precisely this heptad repeat pattern. So if we imagine that we built an HMM that has 7 states to it, corresponding to those 7 non-equivalent positions around the alpha helix, then we could say that there's an A, B, C, D, E, F, G position, and the A positions and the D positions in particular would tend to have amino acid distributions that are biased towards hydrophobic residues. The others, as I say, may have weaker biases. I wouldn't put any faith in these exact numbers here, just made up some numbers, but more or less A and D are biased towards hydrophobics. The others may be more weakly biased towards hydrophilics. And that pattern is going to mean that we generate sequences from this HMM model. They will be different sequences each time, but will tend to reflect those patterns. So as we walk through this model, we might start in position A, so packed in a position where one helix we're generating is packed against the other one, and usually we will pick a hydrophobic residue there. Maybe B, C have weaker preferences, but D we will usually pick a hydrophobic. As we keep going, we would have these various preferences, and if we just keep cycling through this over and over, we will tend to pick sequences that have this characteristic 3-4 repeat pattern of hydrophobic amino acids. And that is going to be a pattern that we can look for to identify that we are looking at something that is likely to be a coiled coil. Now, so far, I haven't shown you anything you couldn't do with PSSMs. And this is a much more difficult mathematical model to work with, so it's harder to think about. The algorithms to fit data to these models are more complicated. The algorithms to learn these models are more complicated. So there must be some point to using them. There must be some advantage relative to a PSSM to make up for that greater complexity. And we can see why that's the case by thinking about some of the ways that maybe real coiled coils would differ from this perfect model. So, for example, in a real coiled coil, it may be that we don't always go from position A to B to C to D to D to E and so forth. So it might be that every so often you've got kind of an extra bulge in your helix, or maybe it doesn't add up to exactly 7 amino acids in every 2 turns. So sometimes you have to kind of adjust to catch up. A PSSM can't really capture that. If you've got a 7-position PSSM, you have to find those 7 positions, and it's going to have a very hard time dealing with a sequence that is almost a match but has an extra thing stuck in it. With an HMM, though, that's a pretty trivial modification. So we could throw in some extra states and just call them these X to represent a kind of generic background state to represent the possibility that maybe we move off of the standard motif pattern. So when we're in ability, we skip to an X and then from X to B. So maybe 99% of the time, or 99.9% of the time, A will be followed by B, but 0.1% of the time, A will be followed by X and then by B. And what we've done by doing this is get a model that can now recognize some flexibility in the pattern. So it can account for the possibility that maybe we have some of these extra amino acids and still recognize the sequence we're trying to recognize. We could go even further and add in what are called self-transitions. So we could say that sometimes X is followed by another X. Again, maybe 99% of the time A is followed by B, but 0.1% of the time A is followed by X. And then maybe 90% of the time X is followed by B, but 10% of the time X is followed by another X. That gives us some flexibility to say that maybe it's not just one amino acid, maybe there's a possibility we have 2 there. Maybe there's even a possibility of a longer loop where the sequence just breaks out of that coiled-coil, goes off, does something else, and then rejoins the coiled-coil a few amino acids later. So that kind of flexibility is something we can't build into a PSSM, we can build into an HMM. We could likewise represent skips, so maybe sometimes you miss the B position, just go straight from A to C or B to D, and so forth. That also is something we can build into an HMM. So an HMM can capture much more complicated patterns. Now, unfortunately, as I mentioned, the computation to deal with these HMMs, so to take advantage of that greater flexibility, is significantly more complicated. And this is unfortunately one of those places where, for this particular class, I'm not going to teach you in enough detail to really be able to do this yourself if you haven't had some prior computer science algorithms expertise. I will say that there is a general class of computer algorithm called a dynamic programming algorithm, which gives us a way of taking a sequence and figuring out what the likelihood is of a given HMM generating that sequence. So with a sequence and a PSSM, we can very easily, by hand, do the math to figure out what is the likelihood that PSSM would have generated the sequence. That's not so easy to do with an HMM, but there are computer algorithms that can do that. At a high level, a dynamic programming algorithm is an example of an even broader class of algorithms known as divide and conquer algorithms. It's a very general paradigm of computer science and computer algorithms that if you're trying to solve something hard that's maybe non-obvious how to solve, you can often try to see if you can take that hard problem and break it down into easier problems, so that by solving the easy problems, it becomes easy to solve the hard problem. And in this particular case, that can be represented in what's called a recurrence relation. And a recurrence relation is basically a mathematical way of saying that the problem of figuring out the maximum likelihood assignment, or the maximum likelihood, or the likelihood of a particular HMM generating a particular sequence of I nucleotide, or I amino acids, so I characters in a row, can be determined based on the likelihood of that HMM generating patterns of I-1 nucleotides, with some other things telling us about the probability of making particular transitions in that model, and the probability of outputting particular amino acids from particular states of that model. So when you can represent a model mathematically in terms of this kind of recurrence relation, you can often come up with these dynamic programming algorithms, where you can then start by looking at your model in terms of very simple assignments, so figuring out the likelihood of generating just the first amino acid of your observed sequence from the model, and then use those likelihoods to figure out the likelihood of generating the first 2 amino acids, use those to figure out the likelihood of generating the first 3, and so forth, until you've built up your complete solution. So again, I don't expect any of you who haven't had an algorithms class to follow exactly how that works, but I want to give you the high-level idea, the kinds of paradigms that go into this. And for those of you who have studied some algorithms, you should be able to figure out from this kind of recurrence relation how you would actually write an algorithm like this. And this is something you would get to in a more advanced computational biology class. But basically, I want you to take my word for it for now that this is something we can do. If you give me an HMM, or you give a computer program an HMM, and you give it a piece of sequence, it can generate the likelihood that that HMM would have produced that sequence. And we can use that just as with PSSMs to run through a library of possible HMMs representing different protein structures, come up with the likelihood of each possible structure generating that HMM, and of course add in prior probabilities just like we saw when we looked in the PSSM case to come up with the maximum likelihood assignment. So the most likely model, whether it's one of our HMMs, or protein structure, or background model, the most likely model to explain our sequence. And again, just a reminder, this is not something specific to sequence analysis, as is often the case in this class. The basic computational tools that turn out to be useful in one problem, so recognizing sequences from a given protein structure, will actually turn out to be a useful mathematical abstraction for many other kinds of sequence analysis problems that may be biologically quite different, but have similar mathematical abstractions. So that basically is what we would do with an HMM. Questions? All right, so figuring out whether a sequence is generated by an HMM or whether that sequence has high likelihood for that HMM is something we can do, but it's more complicated than with PSSMs. The same is true, unfortunately, for learning HMMs. So I could show you exactly how you would build a PSSM from a set of example sequences. That's not something we would want to do by hand for an HMM. If you actually have labeled sequences, so someone has, let's say, taken a bunch of coiled coils and manually, by hand, showed you which were the A, B, C, and D positions, it's not actually that hard to figure out the HMM that goes with that set of data, as long as we know in advance the structure of this. So if I showed you the structure of the HMM, that is the graph with all the probabilities missing, and I showed you these labeled examples of sequences, it's not so hard to go through and figure out what all the probabilities should be. So, for example, you could just look in your labeled sequences at how often a B follows an A, and then get the transition probability of A to B from that. You could look at how often state A corresponds to a lysine or a valine and so forth, and get the probability distribution of outputting different amino acids in position A. So all of that is doable. Usually, though, you don't have labeled examples, especially when you're working with very large data sets. You don't want to go through and have to manually label everything. And there are actually methods where you can infer the probabilities of your hidden Markov models without having the labeling. Effectively, just by knowing that you're seeing sequences that are examples of this hidden Markov model, figure out simultaneously what the labeling should be for those sequences, and what the probabilities should be in the Markov model that correspond to those labelings. That, too, is more advanced computation than we can get to in this class. It involves a technique called the Baum-Welch algorithm. It's an example of a kind of more advanced statistical inference method called expectation maximization. Again, this is something you would learn in a more advanced computational biology class than this one, but for now I'll just ask you to take my word for it that this is doable. There are computer algorithms that can train these models and learn models from example sequences, and those are actually quite important to how a lot of sequence analysis is done in computational biology. So, once we've done this, we more or less use an HMM just like we would use a PSSM. So we can take examples of sequences, we can build libraries of HMMs, and when we actually get into hands-on domain recognition, so trying to find structural assignments for our proteins, we'll be using some pre-built libraries of sequences. Some of those pre-built libraries will use PSSMs to recognize examples of protein structural domains. Others will use HMMs, and I'll point out examples of those when we get to them. There are also others that use other kinds of methods, or some that are based on adapting sequence alignment tools, but basically HMMs are one of the examples of how this would work, and it works very much like the way we use PSSMs. We get our library of HMMs, we get our prior probabilities, and then any given sequence we can run through our possibilities and find the best assignments and figure out whether we're seeing anything that is more likely to be an example of a given HMM than any other HMM or than the background. Questions? Right, so that basically is what I had to say about recognizing protein structures using sequences. And as I mentioned, that it's typically going to be recognizing that a sequence comes from a class of well-conserved protein domain. There are some cases where we want to search for sequences using other sequences, and I'm going to go through that more quickly, simply because the tools aren't as standard, they're really not as reliable, so not something you'll be using as often, but it is worth being aware that this can be done, and there are tools for doing it. In some cases, we want to directly align three-dimensional structures. So we might have a protein structure, and we want to go through a database of known protein structures and ask, is there anything similar to this protein structure in that database? So this might be something we'd be interested in if maybe we're trying to find a good homologue of our protein to figure out whether, let's say, drugs that act on homologue might act on our protein. We might be interested in figuring out whether maybe an antibody that binds to one example of a protein structure is likely to be reactive, so able to bind with our new example of that protein. So those are cases where we might be interested in aligning a known protein structure to another protein structure. So there's one version of this problem called a rigid alignment. And a rigid alignment means literally taking your two 3D structures, pretending these were perfectly rigid objects, and finding the best possible way of lining those two objects on top of one another, and usually coming up with an assessment of how good that alignment is. This is actually a pretty easy problem. I'm not going to go through the algorithm because it involves more advanced math than we're assuming here, but basically it comes down to some relatively straightforward linear algebra, that is matrix algebra transformations. So this is something that can be done efficiently, and is in fact done pretty routinely. It's a core operation in a lot of kinds of structural analysis we might do, and there is something called the Kelch algorithm that will do this for us. Unfortunately, when you start moving to allow flexibility, all the theoretical guarantees go away. There is no standard efficient way of taking two protein structures that maybe are largely the same, but are distorted relative to one another, and reliably figuring out that you are looking at almost the same thing. So we might have these two structures shown in blue and red here, and we might be aware that these two structures are virtually identical. It just happens that there is a bend in one structure relative to the other, and you or I could look at that, maybe recognize that, and figure out that these are almost the same structure. Unfortunately, algorithmically, that is not easy to do. You could, of course, run rigid alignments and figure out that there are big rigid pieces that align between these, but it's not trivial to figure out exactly how similar these are relative to one another, or to identify that they are different by just one small change like that. There are some tricks we can use to bring sequence alignment into this kind of analysis and get things that give us theoretical guarantees and efficiency comparable to what we get from sequence analysis tasks. So one way of doing that is to recognize that to some degree the structure of the protein is a sequence. In particular, we had seen the structure hierarchy, and you may remember that secondary structure, the first step beyond the amino acid sequence, is itself a kind of sequence, or at least can be represented that way. And in particular, a sequence of angles of rotation around the amino acid backbone that we refer to as the phi and psi angles. So you can represent the secondary structure of a protein as a sequence of phi and psi angles, and you can align these sequences of phi and psi angles using algorithms very much like what we would use to align sequences of nucleotides or sequences of amino acids. So we can take any possible alignment of these, and instead of looking up in, let's say, a substitution matrix, how good the alignment of two different amino acids are in a position, what we would do is line up two phi angles or line up two psi angles in the corresponding proteins, and we would score the quality of that particular position of the alignment by how close the particular angles are. So if these two phi angles are 30 and 40, maybe we say that that position contributes minus 10 to the overall alignment. So we could do that for all of the aligned positions. We can throw in gaps. We can use very much the same algorithms as we had seen for sequence alignment and get something that will align secondary structures by treating them as strings of angles. You could also do a discrete version of this, so simply treat a protein as a string of secondary structure elements, so alphas turn, alpha turn, beta turn, and so forth, maybe gaps between them. Again, it's virtually the same algorithms as if we were aligning nucleotide sequences. But of course, there's more to an alignment than the secondary structure. That's not going to really capture what we necessarily want in a full three-dimensional structure of a protein. But all the theoretical guarantees pretty much go out the window when we start talking about three-dimensional structures. Because of this, there are a lot of methods out in the literature, but nothing that will really give us guaranteed quality and guaranteed accuracy and guaranteed runtime. So there are a lot of heuristic strategies that people have brought to this field to try to come up with good, reliable, fast tools for structure alignment. One example that we may see when we get into the real databases is to try to abstract the problem into something simpler that we can work with more effectively algorithmically. So, for example, the vector alignment search tool, or VAST, uses the idea of taking our proteins and reducing them to vectors representing these big pieces of secondary structure, and then more or less throwing away the details of the protein and working with these vectors in 3-space. It's easier to align patterns of vectors. It's a simpler representation than it is to align entire 3D protein structures. So that's an example where by stepping back from the problem, we lose some detail, we lose some guarantees of accuracy, but we can do things much more efficiently and get a practical analysis tool. There are lots of other tricks out there. So some methods use a blast-like strategy, using the fact that we can do rigid alignments easily to construct blast-like seeds of structural alignment, get little pieces that nicely align in a rigid alignment, and then just as in BLAST, use heuristic methods to try to build in flexibility around those seeds, grow outward from the seeds to build full three-dimensional alignments. You may see some tools that work like that. And then there are more exotic kinds of things, such as what's called a contact map overlap approach, in which we look at an abstract mathematical structure called a contact map, and we represent it as a matrix or a graph, which is basically a way of mathematically encoding which amino acids are close to one another in one protein versus another. And just like with the vector alignment, reducing things to this more abstract mathematical form gives us a simpler mathematical object in which it's easier to do more complex algorithms, it's easier to do efficient algorithms, and identify similarities across distant patterns of proteins. But this is really an active area of research. People are still working on new methods. There is nothing that is universally used, like BLAST is almost universally used for protein alignment. There really is no similarly trusted BLAST for structural alignment, at least not yet, but it's an active area of research and an area where things are continuing to improve. And if you really want to do this now, there are tools that can do this for you. I may demo one briefly when we get to these structured databases in the next lecture. But roughly speaking, that brings us to the summary of what we've covered today. So we have looked at searching and alignment of protein structures, in particular searching structured databases using either sequences or other structures. It's worth knowing that there are good, well-accepted methods for matching protein sequences to protein domains, so taking an amino acid sequence and finding structural patterns that may correspond to it. These methods are widely used for this problem, and they have other important uses throughout sequence analysis, so it's good to know something about these methods and how they work. Matching structures to structures is doable, but it's a much harder problem. The methods are not as well developed. There aren't simple standard things that everyone uses, but this is something where the tools are getting better and better, and if you want to do this, it's worth knowing there are tools out there that can do this for you. So any questions about any of this? Okay, I guess we will break for